I cannot believe this.
The steam makes it.
No Broncos.
Yeah.
It's right in here. Your tattoo. Number 18 tattooed right there. You guys are not going to believe this. I have all of the PowerPoint for this section of the course loaded up online.
He's like 40.
Here's the deal, those of you watching at home also. I have received your massive swamp of emails for your primers and such. I have not had a time to process through all of those to get all the primers in the right format so that I can paste them all and order all the primers from IDT. Which means next week I will not have the materials you need to run your laboratory stuff. Which means I don't know what we're going to do next week. So who wants to take a break? Who thinks we should meet anyway? So if even one of you wants to meet, great.
Not even in this class.
We're all gonna okay. So,
I'm not voting, I just have a question.
Yeah.
Out loud. Do we just have one lab report due at like the end of everything? Or like
Yes.
what's the lab report format?
Yeah. So hopefully you give me a tube that has your plasmid in it, and then you'll need to do a write-up. You get to do a write-up
Okay.
about all the activities, and I will give you a format that will allow you to say, oh, here's his cDNA sequence and how he wants it organized. So you can lay all of that stuff out and you have a nice organized printout that you give to me with all of the data that you've accumulated the past 3 weeks.
Okay.
And I need that in a standard format if I'm going to make any sense of it. I could spend probably a whole week trying to figure out, because you would all organize it differently. And it would just be murder, and I just don't want to do that around
Right.
finals.
Okay.
Thank you for asking. Yes?
Will you be doing any selective fighting once we transform?
Yes. So you'll be using ampicillin plates. And so the bacteria that pick up your plasmid will be resistant to things that are things that haven't picked up plasmid won't grow because it's a laboratory strain of E. coli that is super wicked. Ampicillin is a drug you can't use anymore because it's not effective against in the natural world, but in a laboratory setting, we have an artificial environment. Okay, so so I keep unpinning this and it keeps coming back. Is that Google trying to tell me something?
Mine does it too.
It's a virus.
Chromes. Yeah, 10 chromes on the bottom. and
If you thought the US government was invasive, that's what it's telling me.
Oh, dude, my sister was picky. Oh shit, I've only gotten to 34 and I was like,
Isn't this cool to see it on this side? You've never seen this stuff.
Okay.
Oh, I can delete this one.
Alright, I'm getting sick of Candy Crush.
No, get plastic.
I had to get rid of it while sitting for that.
Yeah, delete them, delete all of them. I deleted
Okay.
my Facebook.
Last time we left off, and we hadn't yet gotten through calculating, so we learned, 1 to 2 to 1, 3 to 1, 9 to 3 to 3 to 1, and we hadn't gotten into real specific details. So I'm going to throw a couple of examples up here. In the PowerPoint file, there are examples that walk you through it, but and of course we're recording this, so you can you can go through this again. When I was in your shoes, I took a course, and the professor said, if I cross two I want to know, I want you to tell me how many of the offspring are and I went, oh man. And I drew this, I flipped over the page and I drew this enormous tetrahybrid cross and kind of did some rough guessing. And yeah, it was a multiple choice question, so there's an answer. I'm like, well, based on what I'm doing partway through, because I'm going to spend most of my hour doing this, I'm going to guess the answer to something. There must be a different way to solve it, because it's an hour-long exam, and I'm not supposed to spend 45 minutes drawing this stupid tetrahybrid cross out, right? So the way you do this is you can actually just get a ratio for each individual trait. So I just set up this cross here. All right? I guess I should just do it as a box. Hi, I'm awake.
You're bored, like you're not a good box. All right, I was not happy.
Did that scare you? I promise I didn't touch it. I don't think I touched it. Okay. So the odds here are 2 out of 4, right? And then B is going to be, well, these are all capitals, and this is big B, little b, and I'm looking for a big B, little b, right? So I can, big B, that doesn't work, big B, that doesn't work. So this is 2 out of 4, right? So if you just draw a monohybrid cross for each trait individually, I could take this out 7. And you just wind up with an equation, and then these are just multiplied together. Right? So this is going to be, this actually is 2 to 4 also. This is 2 to 4. I randomly wrote them up, and I randomly wrote them up. Right? So it's going to be half, 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 so with that 1 over 32 or something like that. 1 out of 32 offspring will be this genotype. Do you guys understand that? That's pretty simple. Let me mess it up for you now. I need to choose a different one because this is dark enough they might not be able to see it there. And I want the offspring. I'm not interested in the genotype of the offspring. I'm interested in the phenotype of the offspring. Why do I have empty spaces there? It doesn't matter. If what I'm looking for is the dominant. It doesn't matter if this is dominant. Oops, sorry. Dominant or recessive. It looks dominant because I'm only looking for phenotype, right? So now when I do my monohybrid cross, what I get is 3 out of 4, right? So I can work that out. I can work out each monohybrid cross. Wow. So for this one, it's 100%. So it's 4 out of 4 for B, right? For C, what is it? 2 out of 4. So I can draw these up in my head fast enough. I can just look at the cross and figure it all out. So let's do the C just so we go. Right? So it's those 2 out of 4, right? And then D is going to be 2 out of 4. What if I went and the reason I wrote this instead of a dash in here is the only way I'm going to get the recessive phenotype is if they're both recessive, right? Okay. There are a couple of examples in the PowerPoint file that you can work your way through. If you go online and you type in tetrahybrid cross or find worksheets of these things on there, genetics worksheets and such. Okay? Guaranteed on the exam, right? Introduce one word to you, which we'll get into more detail later, is a pedigree. The word pedigree basically refers to a standardized format for drawing out multiple generations and representing all of the individuals in that generation. This looks to be like a human one here, where we have I think that's female and that's male. Normally they would be reversed that way. We have a female and a male. If there was, if you go back in time, maybe there was a child and the child died early on, or there was a boy or a girl. You go way back in generations. They'll draw. They don't know what it was, right? So here here's a couple. They mated, and they had this child, and they had this child, and then a child came in and married. And there's a hash mark across them because these people are deceased. People that are alive and you get samples from do not have hash marks in them. So this person married into the family, and they had 1, 2, 3, 4 girls and no boys. Right? Someone married in. This, ooh, that's kind of creepy. That's cousins marrying. No, an uncle would be. An uncle would be like that. Yeah. So this is like a first cousin, second cousin thing. First cousin. Yeah.
Isn't your parents' cousin your
It happens much more frequently than you would know, than you would expect. And families disperse, and the cousins lose contact, and then kids wind up in college together or something like this, and they discover that they're related later on. There's at this point, ideally you'd want this to go down another generation so that you get even more genetic mixing. This is probably okay. This, do you know why it's not good for humans to be inbreeding like this?
We'll get hip dysplasia.
Hip dysplasia, that's right. And nobody wants hip dysplasia. Now, we've removed ourselves from natural selection for so long that we have accumulated mutations in essential genes. And the reason that we can survive is we get a copy from mom and a copy from dad. If a copy from mom is bad and a copy from dad is good, we can still survive. But if I've got a copy from my mom and that traces down and that copy has been going down into my cousin, and then I have kids with my cousin, we could get the 2 bad copies together and we get a kid that has some challenges. Crossed eyes, hip dysplasia, PKU,
Oh no, I have that.
diabetes.
I have PKU.
You have PKU?
Yeah.
Oh, really?
Yeah.
Okay.
I don't think my parents are coming.
100 years ago, you wouldn't be here at Mines. Wow, so you got to watch your diet real careful, huh?
Yeah.
Oh, sorry to hear that. Good for you. Okay, so this is basically this Huntington's disease. Just showing the family pedigree here. Okay, let's talk about extensions to Mendel. Situations where Mendel doesn't work right. And we're going to talk about, I'm going to break down Mendel's laws. If you look at it superficially, Mendel's laws fail in an easy way, and then they fail in a way that's really, really messy and complicated and requires a lot of bullshitting and stuff. Hey, it must be like this. We don't know how what's going on, right? Let's talk about the easy ones today. So, we use the word wild type. Wild type is used a couple different ways by biologists. But you probably have heard of things as, if I have trait A, that's not good. I can't use that right because it just isn't dark enough. If I have trait A, if I have these heterozygotes that I cross, I could get these possibilities, right? You know this. And so since A is the dominant, most people would use the word A is the wild type, right? Some people use wild type to mean dominant, and that's incorrect use of that, although it's almost always true.
Thank you.
There are circumstances where there's a rare mutation in humans that is dominant. So if you inherit the Huntington's mutation, for example, or Parkinson's, if you inherit that mutation, it expresses itself, it's dominant, but it's very rare in our population. So it's not the wild type, it's not the predominant form of the gene, right? That's what wild type means. The one that occurs most in the population. That's how people generally use the word wild type. The strict definition that someone came up with and that we're all expected to adhere to is if a variation is in the human population 5%, if it's in a population, 5% or more is called wild type. So most people would say that in the human population, brown eyes are wild type because most people have brown eyes. It's the most common form of eyes. Blue eyes appear more than 5%. Blue eyes are also wild type, technically, in the human population. Does that make sense? Okay. So, you surely understand that there are green eyes, and hazel eyes, and blue eyes, and brown eyes, many different colored eyes. So there are some people who have red eyes. Did you know this? They have no pigments, and so what you see is the blood. It is possible if you would so like. Oh man, it was maybe 10 years ago. There was this guy out in California who said, I can turn your brown eyes blue. And he would do a laser treatment. What he's doing is destroying the pigment cells so that there was less brown pigment, and so the eyes are blue. And then eventually the blue was going away too. And we're left with red, like bloody eyes. Not good. Anyway, it's not like they were oozing blood out, but they just didn't have a color there anymore. It's not good.
Why didn't you just wear colored contacts?
Does that make them like super sensitive to light though?
Yeah, it's not good. Much more prone to eye diseases. Okay, so a polymorphism. There's many polymorphisms of eye color. Poly, many, morphism is form. Many forms of eye color humans. Who's got hitchhiker's thumb? How many of you can bend your thumb way the hell back like I can? It goes up and then how many of you bend your thumb? There we go, right there. Your thumb is straight up. And that's so actually, both. So there's two polymorphisms for hitchhiker's thumb.
You got number one.
So I am homozygous recessive. I have hitchhiker's thumb. If your thumb stands up straight like that, you can't bend it back and angle it the way I do there, you are one of these alleles. Both are wild type, but the non-hitchhiker's thumb is dominant, right? Okay. It turns out, when we're talking about hitchhiker's thumb, this particular trait, either you have hitchhiker's thumb, does that look gross to you guys out or what? Either you have hitchhiker's thumb or you don't have hitchhiker's thumb. There's no in between. And since the non-hitchhiker's thumb is dominant, it's actually completely dominant. So some of you that have the non-hitchhiker's thumb, like this, you're carriers, right? Some of you carry the recessive version of this. But we don't see it, there's no in between, right? So non-hitchhiker's thumb is completely dominant over recessive hitchhiker's thumb. So when Mendel was looking at round peas or wrinkled peas, to his naked eye, he was looking at the seeds, that one was completely dominant over the other. If you take a microscope and you crack these things open and you look at them, you can actually see a difference between homozygous dominant, homozygous recessive, you can tell just by looking on the outside. But the heterozygote looks to the eye like the homozygous dominant. If you crack it open, there's differences to the way the structure is internally. Okay? So there is an in-between, yeah.
So like, I don't know, like I know like, I think it's like a fourth of the population has hyperextended knees. Is that something kind of like your weird thumb that goes too far?
A quarter of our population has hyperextended knees and what?
Like, is that something kind of like how technically like a quarter of the population should have hitchhiker's thumb? Is that something that can be done?
You know, the ratios depend, there was probably some evolutionary selection to why things were a certain way.
Okay.
Blue eyes have some advantages over brown eyes, and they have some disadvantages. And it just depends what the environmental selection was. And then, of course, humans are selecting beyond it. I mean, many people would say that blue eyes are, oh, he's a hunk with blue eyes, and so for some reason it's a more desirable trait, and so it stays in the population when maybe it ought to be weeded out, because blue eyes probably are less advantageous than the benefits that come from it. So I don't know. I kind of went around your answer there. But
Yeah, I don't know, because I feel like with your knees,
Just-
it's like,
yeah, just because
you can either hyperextend them or you can't.
Yeah, things don't necessarily exist in these. Just because it's a 3 to 1 ratio when
Yeah.
you do a cross, doesn't mean it'll be that way in the population.
Yeah, I was just wondering.
Yeah, yeah. So we can find things that are incompletely dominant. And this is where the people breeding the lambs and the ewes together probably got their idea of blending. Is the people who were doing the breeding experiments back then were not going, they would breed and get the next generation, they wouldn't pursue to the F2. So they only went parental to F1, and they didn't look at the F2. That's what Mendel did that was different, is he followed it many generations. So if you take this red, what is it, a 4-o'clock fob flower, and a white if this is true breeding and this is true breeding, and you breed them together, all of the progeny are pink. Tomato soup, cream of mushroom soup, mixed them together. And if you don't follow it, you'd say, oh, they're permanently and forever mixed together. Now I have pink, if I cross pink with pink, I'm going to get pink because I'm more blending, right? That makes sense. But when you actually cross it out, what you get is 1 red, 2 pinks, and 1 white in that ratio. So this follows Mendel's laws. You segregate these things back out, and you actually get the parental phenotypes back in the next generation in the 1 to 2 to 1 ratio. So blending does not occur. It's only because you've got an intermediate phenotype. So this is called incompletely dominant. So the reason this works is, in this particular situation, the dominant allele, which we'll just say is A, well, I guess it would be R for red, right? This codes for a protein. And this protein folds up in some shape, and it takes some non-colored compound, and I don't know, it probably puts like a, some functional group on, like an OH group or something on it. So it takes it, it's an enzyme. You take a colorless compound, and you get a colored compound. You get red compound. And what's happened in the little r, things that inherit the little r DNA, probably, so this has your messenger RNA, and you've got your ATG and your termination codon, so you've got a great protein coding sequence here. In the little r, what you get is maybe your ATG goes and hits a termination codon very early on. So you don't get the rest of the protein made. So what you get is that, and this is not a functional enzyme. So what your colorless compound can't come in and get modified into a red compound because the enzyme is dead and doesn't work. So in many cases, the reason you have a recessive allele is the recessive allele is screwed up and doesn't work anymore. People have blue eyes because the people who have blue eyes are missing an enzyme that makes the pigment darker like it should be. Right? They just have less pigment. So in this case, an organism which is a heterozygote, it's a carrier here, big R, little r, it has some enzyme but not all that it needs to be red. So it makes a little bit of red so it comes across as pink. Okay? Questions on that? Okay. So that is incomplete dominance. I'm going to talk to you now about codominance. And you'll need to work through this. You need to understand the difference between these. And I test on these, specifically these concepts, your ability to understand the difference between incompletely dominant and co-dominant. People mix them up a lot. Incompletely dominant means you get an intermediate color, something that's in between the two. You'd have something in between hitchhiker's thumb and non-hitchhiker's thumb. Co-dominant, ooh, that doesn't work. Great. I'm gonna call it A. Oh, man. That suck. See? Geneticist, not basketball player. I'm going to call it A prime right now. If I breed a pure breeding A with a pure breeding A prime, I get a heterozygote. And what I should get is something that is both A and A prime. Here's a classic example of this. You guys have seen this. I have a flower which is red, and I have a flower, I'm looking down on the flower, right? And I have a flower which is yellow, let's just say. Same species. This one's just red flowers, this one is yellow. When I cross these guys together, right? When I cross these guys together, I wind up with a flower that is red and yellow. You guys have seen those before, right? Are they petunias? Do that really commonly?
I've seen it.
Some kind of flower, right?
Flowers exist.
Geranium, maybe a geranium does it. You guys understand it's striped. It's red and it's yellow. It's both parents at the same time. And so red is not dominant over yellow, and yellow is not dominant over red. They're both dominant. They're co-dominant. They can share. Right?
It happens in orchids a lot. That's where it happens.
Happens in orchids a lot? It could be. The classic example of this is blood typing. And let me explain to you the molecular biology behind blood typing. So here we have a red blood cell. We're going to zoom in on the plasma membrane of the red blood cell. And there is a protein called, what's it called, H factor? H factor. It's called H factor. And onto this protein, sugars get stuck. And they get organized in the A fashion. So they have a particular three-dimensional shape. So you have a unique set of covalent bonds to connect those sugars in that way. Onto those H factors, you could assemble the sugars a different way, and those covalent bonds will have a different three-dimensional shape. And so they look different. They're a different molecule because they have a different three-dimensional shape. So if a person is A and A, if they have the enzyme that connects them in the A format and the other enzyme that the person has connects them in the A format, that person has A-type blood. I could inherit from my parent the enzyme that puts them together in the B format instead of the A format. And from my other parent, I inherit a B. So now this person is A, and B, B. And the progeny from this cross will be AB, which means on the red blood cells, so the person in their body has the enzyme to make things put on in the A format, and they have the enzyme to put things on in the B format. And so the body can do both, and their body does perform both. And so their red blood cells have both A shapes and B shapes on them. Right? Yeah.
How are the negatives and positives introduced?
That's a whole nother system that's unrelated to AD. It's a totally separate gene. Okay, we can get to that later. So A doesn't dominate over B and B doesn't dominate over A. They're co-dominant, right? We can mix this up by adding. So here we have, if I draw this out, basically what we have in this situation is this labels them in A pattern. This adds in B pattern. Oops. And the reason this enzyme is different, it probably folds up in almost an identical shape. But this guy probably has a few mutations along the way that change the shape subtly so that the covalent bonds get connected a little differently. Does that make sense? We can actually have another allele where potentially we make our screwed up enzyme. So in this allele, non-functional. In this allele, this enzyme can't put sugars on a red blood cell. So what we've done, rather than mix things up, because if I do this, you can't take gene A and gene B and cross them together. So we actually have to make a common system. So we actually call it the I allele, and I don't know why it's I. There's the A version and the B version. Like that. And it's capital because A is dominant, and it's capital because B is dominant. In theory, I could have a person who's recessive, who has an enzyme that's dead that can't put sugars on it. In which case the progeny would be this. And they would be A, it would look A. Now, that gross morphology, just looking at it, let's see if I can do this. So this person has 4, we'll say, 4 H factors here, and it's got the A sugar on here. In theory, this guy should have half the number of the sugars on here. Half of these should be unloaded, right? If you went with a microscope and looked at each individual protein, I bet you'd find that. But we don't see it because we're just looking at a large, big picture. We don't zoom in close enough to see that, right? So this guy is phenotypically A. Right? So we have 3 different alleles. We've got IA, IB, and little I. So A and B are codominant to each other. A is completely dominant to little I, and B is completely dominant to little I. Right? So, you guys ever heard of this fellow Charlie Chaplin? The embarrassing thing is that's on YouTube now forever. So Charlie Chaplin was blood type O. What does blood type O mean? Recessive. Little I means he didn't have any sugars on the outside of his blood. He had a girlfriend, he never married her. I'm trying to remember her name, Joan maybe? Joan is blood type A. What is she?
Either she's
One or two. So this, and then we don't know what that is? Yeah. Okay. So Joan had a kid and said, my ex-boyfriend Charlie Chaplin is the father, and he needs to pay my medical expenses, and he needs to pay child support. The child is blood type B, what is the genotype of the child? What's this? We don't know, right? Just from that description. So, from Charlie can only give a little I, so it has to be a little I, right? So she had to get the B from her mother, this child, and so this must be this, right? Then she would be blood type A. She's blood type A. So she can only be this. So if the child is this, then Charlie must have given the B.
Or somebody else. Maybe that's why I never married.
Alright, so you can see, this is in the 1940s. And this is all dicey. People didn't quite understand how all this stuff happened. And so the judge said, the jury's getting confused, I'm throwing that evidence out. Jury, what do you decide? Well, she said he's the father. They were dating, so pay the child support. Charlie Chaplin was found to be the father of the kid. Of the jury. Okay. This type of analysis, when I was a kid growing up, blood type was evident. They would look for blood type at the scene of a crime. It was important in determining paternity. Now we actually look at DNA sequence. We go down, they do PCR, and they do something we call restriction fragment length polymorphism, RFLP. We'll talk a little bit about that at the very end of this section here about the DNA analysis.
What exactly happens if an individual with A-type blood is given B-type blood? Does their body just reject it?
So let's talk about that. I don't have it here. Okay, yeah. So if a person has type B blood, it all comes down to immune response. So if I'm type A blood and you're type A blood, if we take your type A blood and put it into me, my body says, yeah, type A blood, that's what I am. You just put me into me. We're okay. If I have A and you have B, if we take your blood and put it into me, my body says, the shape of B, these sugars on here, the way they extend off this way, that is an invader, and I need to kill it. And red blood cells are really fragile, so the immune system, when it attacks it, the immune system will rupture all the blood cells and essentially coagulate the blood all throughout my body in a short amount of time. Generally not a healthy thing for keeping oxygen flowing through these systems here, right? So a person who's type A blood can only receive type A blood, or it turns out, if you have no sugars on, if you just have H factor with no sugars on it, like that, your body says, oh, it doesn't have a shape, so there's nothing to attack. Okay, cool. Type O blood, come on in, we're okay with it.
Would have been added to type of whatever sugars
No,
now.
no,
Doesn't bother that.
no, it doesn't bother. So a person who's type A can receive type A and can receive type O. If a person's type B, that person can take type B. They can also take type O. If you're type O, can you take type A? No, you're dead. How about type B? If I'm A, can I take AB blood? No, B is not self. I'll destroy all the stuff that's put in me and clog up my veins, arteries. Yeah.
Random question, but is like if you're doing a transfusion, is there any way you could change your blood type from A to O? Like if you like cycled it out, you know, like
You'd have to irradiate the person and destroy the entire immune system.
okay.
It's like even just having variation in this, because
Why would something like this evolve in humans?
yeah, because it can't be for blood transfusions. That's modern technology.
I don't have an answer, and I can't pull an answer out of my butt either right now. You normally I can, but you stumped me. It's not often I get stumped. So, a person with type O is a universal donor. Who here, does anyone, are you type O? So if you believe in donating stuff, every time you are eligible to donate, please do not. Please donate. Right? Please donate. And the reason is, they come wheeling someone into the emergency room and someone's been shot and they've lost a lot of blood. They can't go, okay, take some of the blood, run it off to the lab, we'll be back in 15 minutes and let you know the blood type, the person's dead. When they roll someone in like an emergency like that, they have type O blood sitting there. And they grab that type O blood and they put it in. Because everybody can take type O. Okay? They're the universal donor. The universal acceptor is an AB person. If I'm type AB blood, and someone has A blood and they give it to me, my body says, yeah, I'm A and I'm B on both. Yeah, I'm doing it. The universal acceptor is a person who's got AB blood. Good. Yeah?
What's with the positive and the minus? And
The
adding
positive and the negative, it's another gene. There are about, so we talk about A and E, and then people say, oh, I'm positive, I'm negative, or that's a different gene. It's called the RH factor. It's called for rhesus because they identified the stuff in the rhesus monkeys. And it's a separate gene, and it's not as important as this one. There are at least 30 different things on the outside of red blood cells that determine whether it's compatible with you or not.
So if you were like A positive, can you only get A positive? Or is it just A?
It's less than ideal if you take A negative,
Okay.
but it's probably not going to be the end of the world.
What could happen?
Your body would attack it and would clog up your blood.
So it's still,
And I don't know how robust the response is, but the AB is like critical.
yeah.
Okay, cool. Okay, many human diseases. Here's someone with PKU. Phenylketonuria. The person was unable to break down phenylalanine, and it builds up to toxic levels and causes mental retardation if the person eats a normal diet, because there's a lot of phenylalanine in our food. So a person who has PKU needs to avoid eating, you don't eat beans, I'm guessing. Yeah, that would, yeah, you would not be a Mines if you had eaten a diet full of beans. Beans have a lot of phenylalanine and it'll build up to high levels. So she's avoiding, Erica's avoiding amino acid foods, which is, wow. It's crazy. You're eating a lot of like high fructose corn syrup.
Oh yeah, all the like Cocoa Puffs I want.
Heaven! Right. But if the person just avoids eating a lot of amino acid foods, they don't build up the amino acid to the toxic levels, and that's great. Sometimes, and we'll learn about this more, temperature-sensitive mutations. There are organisms, their phenotype changes when environment changes. So this is the Arctic fox. And when it's cold out, the enzyme doesn't fold properly. So the enzyme can't make a colored compound. But then in the summertime, when it's warmer and the body of the organism is warmer, the enzyme folds into the right shape, and so the colored compound can be made. So if you took an Arctic fox and you wrapped a warm wrap around its leg and left it there for a period of time in the middle of winter, the rest of its body would be cold and it would have white fur. When you took this thing off, it would be dark, because the enzyme there in that area would have folded. Isn't that cool? We'll learn about temperature-sensitive mutants. In the blood type, there's also this thing that we talked about. So there's a hemoglobin molecule, this is what carries the oxygen. There is a single nucleotide difference between normal and sickle cell genes. One single nucleotide difference, and it causes one amino acid to be different in the sickle cell than from what we say the wild type. And the effect is, someone like me that doesn't have sickle cell anemia, my hemoglobin kind of packs in kind of an ordered, sensible manner into a red blood cell. in the people with hemoglobin. The hemoglobin is related to tubulin, for those of you who took my cell biology class. Tubulin assembles into long filaments, these are the things that pull the chromosomes apart. And instead of assembling into these globular kind of structures, they assemble into these tubes, and they distort the shape of the red blood cell into something like this. Now this is soft and smooth and squishy, and if it comes to a capillary, it needs to squeeze through, it can squeeze its way through the capillary and get through. This is kind of jagged and it will get stuck, and it will block the capillary. So people with sickle cell anemia, they'll get, have you ever like woke up and you were like sleeping on your arm and you're like, oh, pins and needles, it's killing my hand, ow. People are going through an episode where their cells are sickling on them like this. They'll get all their capillaries all over the body, in their extremities especially, clogged up, and they'll get that tingly, painful sensation. The circulation can be cut off to the point that the parts that are cut off die. The cells that die, and they need to have little bits and pieces taken off. That's not a good deal. Why would such an awful thing exist? You say like, that should be naturally selected out, not good. It turns out malaria is a parasite, it's a protist, and what it does is it goes into these things, it burrows in and says, ooh, hemoglobin's yummy, yum, 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 yum. It eats them, it's totally fine, and reproduces and ruptures out and infects other red blood cells. What happens is if the malaria parasite is in here when this thing sickles, as this thing passes by the liver says, well, that doesn't look right, I gotta take that damaged red blood cell and throw it away. And so the parasite gets destroyed in the process. Whereas this guy, in a healthy person like me, that parasite will pass through by the liver because the red blood cell was. So it turns out, in areas where malaria exists, it's actually advantageous to have some of your red blood cells do this, like half of them. And half of them are normal like this. You have a little bit of sickling and not all of it is sickling. And the effect is it causes the liver to turn over the red blood cells more rapidly, and so the malaria parasite stays at a lower concentration in the body and most people survive. People who are heterozygotes for this have an advantage in areas where malaria is indigenous. In some parts of Africa where there's a lot of malaria, it can be as much as 20% of the population. 20% of the alleles in the population are sickle cell. Because the person who's a heterozygous survives. A person who is homozygous recessive is this from dad and this from mom. In those parts of the country, in parts of the world, because medicine is so not so unavailable for those people, if they live to 12, that's pretty amazing. I mean, in general, the parents realize it when they're little kids, and they're like, okay, well, this is not going to make it. Let's work on a different kid. Let's invest in other kids. This one's not going to make it. If you're a heterozygote, you're likely to make it to 40. There's a really famous quarterback for the Nebraska Cornhuskers, who was sickle cell. He was a carrier. He's probably the best player at the time when he played. He's, in my opinion, the best player in college football. He couldn't get a college, he couldn't get a career as a pro because of this challenge. I mean, if he had an episode on the field,
Was that Tommy Frazier though?
Tommy Frazier.
Okay.
I wonder what he's doing these days. He was
Well,
incredible.
he's a pretty big guy now. I've seen him in a couple camps.
Okay, so we'll end on the heterozygote advantage. Have a nice weekend. As soon as the camera is off,
They kind of like break off naturally.
Somehow he did.